Hello, today we will be doing a presentation on human gene therapy. So what is human gene therapy? Human gene therapy is known as the modification of human genes to prevent or treat diseases. So, how is genetic modification related to human gene therapy? Genetic modification is related to human gene therapy as in order to perform the procedure, the affected human genes need to be modified. In order to modify the specific gene, a disabled virus, called a vector, is used to spread the specific good gene throughout the body, like a normal virus would. However, when it enters the cell, it only transfers genetic material to the cell in need. This genetic material will either code to add a new gene, or replace one, or to disable one. An example of this is called gene targeting. This example involves a homologous recombination of a gene to enter a cell. Once it enters the cell, it can either remove or add genes within the cell. So how is human gene therapy performed? The first process in which human gene therapy could be performed is called ex vivo somatic gene therapy. In this process, bad cells are removed from the body and modified by a vector. Then, once the wanted genes are inserted into the cells will be reinserted back into the patients via their bloodstream. The second option in which human gene therapy could be performed is called somatic gene therapy, in which instead of having the cells being removed from the body, the vector will be directly inserted into the target tissue. This method is useful in curing areas that are directly involved in the disease, such as cystic fibrosis. So what would be some advantages human gene therapy would give to society? Well, first of all, human gene therapy could obviously cure diseases from patients that are currently suffering from them. Gene therapy could also aid in research to overcome other diseases such as cancer and HIV/AIDS. Gene therapy can also silence hereditary illnesses the patient has so that they won't be passed on, and it will provide a new field of work and research for society, thus giving new jobs. Now, what are some disadvantages human gene therapy has to society? Well, it would prevent diseases which aid in maintaining population control. It could also be abused throughout society, as in being only available to the rich, which could result in a civil disrupt. It could also potentially damage the gene pool and result in a lack of variation if certain genes are only passed on. It could also hypothetically result in a mutation of the vectors to result in a harmful virus. And it could also result in a civil and ethical disrupt if it is eventually used for designer babies purposes. So, what is the history of human gene therapy? Well, the first thoughts of gene therapy occurred in 1972 in a paper written by Theodore Friedman and Richard Roblin titled Gene Therapy for Human Genetic Disease. It was then that extensive research in this topic was undergone, till in 1984 a vector system was designed that could efficiently insert foreign genes into mammalian chromosomes. Then, in 1990, the first approved gene therapy took place in the US, under William French Anderson. In it, 4-year-old Ashanti De Silva received treatment for a genetic defect that left her with ADSCID, a severe disease. The effects were successful, but they were only temporary. Currently, gene therapy is developing, and there are clinics and research facilities all across the world that are beginning to be created. Currently, gene therapy is in its clinical trial stage throughout the world, meaning that it is used in research and test purposes for only a select few patients, specifically to what researchers are studying. There are many regulations and guidelines which organizations such as FDA and EMA have set regarding human gene therapy, such as how the vectors are cultured, how the procedures are performed, and how the side effect risks of the operations are assessed. Canada is part of the Human and Genetic Therapies Directorate, which has similar regulations. Gene therapy research clinics are present in Canada, but few, and have been conducting trials. The most recent one, in Edmonton, attempted to cure patients of choroideremia, a disease that slowly turns people blind, and it saw positive results, according to patients. I believe that human gene therapy can be beneficial to society. However, only in the field of medicine, not in the creation of designer babies or modifying physical traits. That being said, I also feel like humans should make sure the study of gene therapy is not too rushed, so that accidents could be prevented, such as the death of Jesse Gelsinger, who died of immune system failure after the effects of gene therapy in 1999. Human gene therapy has, without a doubt, the potential to be one of the most revolutionary inventions of mankind. However, it is important that scientists and ethicists assess the situations in which it should be used that would not spark ethical debates.